So in this lab, we are going to look at fungi. So mycology is the study of fungi, and a mycologist would be somebody who studies fungi. Now, if you remember back to chapter 1 in lecture, we talked about different types of organisms. And you might recall that we talked about fungi. And so included in fungi would be yeast, molds, and mushrooms. And in this lab, we're going to focus specifically on yeast and molds. We're not going to talk about mushrooms in the purpose of this lab. So what I want to do is I want to start with talking about characteristics that are found in all fungi. And so for all fungi, we have either prokaryotic or eukaryotic. So if you remember back to when we talked about this, are fungi eukaryotic or prokaryotic? And the answer is that they are eukaryotic, which means they have a nucleus. If we think about our prokaryotic cells, that would be things like bacteria, for example, archaea, etc. But fungi fall under being eukaryotic. They're cells that have a membrane-bound nucleus. In terms of fungi, they have a cell wall of chitin. And so this is in contrast if you think about bacteria, for example. Bacteria have a cell wall, usually, that is peptidoglycan. Peptido, remember, refers to protein. Glycan is sugar. If we talk about fungi and they have chitin, that is a polysaccharide. It's many sugars linked together. Next, let's talk about how they get food, right? And so if you think about the types of environments that you think of molds, for example, living, typically we think of dark, damp places. And so what that tells you is that if they grow in a dark, damp place, that those fungi are non-photosynthetic. So we'll put dark, damp places. And so if they grow in dark, damp places, that tells you that they're not likely to do photosynthesis because photosynthesis requires sunlight. So fungi are non-photosynthetic. Most are saprobes. And these are microbes that feed on dead or decaying matter. So they're not all saprobes, but many are. And so again, if you remember back to chapter 6 in lecture, we talked about what the term saprobe means. And again, microbe that feeds on dead or decaying matter. In terms of fungi, some are parasitic. Oops. Some are parasitic. Right? And so they can cause disease. When you think about fungi, though, actually a majority of fungi are not pathogenic. They don't cause disease, but there are definitely some that do. And so we're going to talk a lot more about the ones that are medically relevant. So what I want to do first is I want to compare and contrast yeast and molds, meaning how are yeast and molds different from one another? So if we think about yeast, yeast are unicellular. And what that means is they exist as one single cell. So each cell is its own organism. That's in comparison to molds, where molds are multicellular. Multiple cells make up an organism. And so that's a big distinction between yeast and molds is yeasts are unicellular and molds are multicellular. In terms of the way that they grow on a plate, the way that they grow on agar is going to be different. If we look at yeast growing on a plate, the colonies resemble bacteria. They're very smooth, they're kind of shiny. They look very much like colonies of bacteria on a plate. If we look at molds growing on a plate, the colonies have a velvety or kind of fuzzy appearance. So think about, like, for example, molds, think of bread mold, right? When you get bread or when you get mold on the bread, it's kind of that fuzzy appearance. And so same thing, when it grows on a plate, it's going to have this kind of velvety or fuzzy appearance. So you can often tell by looking at the plate whether it's a yeast or a mold. Not always, but often. In terms of reproduction, so yeast, for example, can reproduce by what's called budding. And so what that means is here is this mother cell, here is this daughter cell. And so what happens is that this mother cell is going to replicate its DNA, and a small little piece is going to break off and give rise to another cell. So it's an asymmetric division. When the cell divides into two, at the beginning, it's not dividing into two equal cells. It's dividing into a smaller part that will eventually grow and become the same size as the mother cell. If we look at the way that molds reproduce using spores, and either asexually or sexually. And so again, asexual means without a partner. The offspring would be genetically identical. In the case of sexual, right, sexual reproduction involves a partner. So two molds would come together to reproduce. And so that would be a type of sexual reproduction. Now, for molds, the other thing that we need to talk about is the reason that the colonies have this velvety or fuzzy appearance is that they have these structures called hyphae. And hyphae are these filaments that are used for nutrient absorption. They are filamentous, they're thread-like, and so because of these hyphae, that's what gives these colonies this kind of velvety or fuzzy appearance. And so this is just a simplistic way to compare yeast and molds. And so again, the big difference being yeast are unicellular, molds are multicellular. And so let's look at some examples of these types of organisms. So the first one that we're going to talk about is a medically important yeast, and it's going to be Candida albicans. Now, you'll hear this pronounced a variety of different ways. I've heard it Candida albicans, Candida albicans. It just depends who's saying it. One is not necessarily right and the other is wrong. It just depends who's pronouncing it. So I'm just going to refer to it as Candida albicans. Candida albicans is the cause of a yeast infection, what we call candidiasis. Iasis is disease. So candidiasis is going to be a yeast infection. This can occur in several places throughout the body. One would be in the vaginal area. So if you are female, for example, and you've ever taken a broad spectrum antibiotic, remember that a problem with a broad spectrum antibiotic is that it not only kills the bad bacteria, but it also kills the good bacteria as well. And so it gets rid of some of your normal flora. In the vaginal area, you might have Candida albicans and a variety of other bacteria that are beneficial, right? And they're there to serve a purpose. If you take a broad spectrum, though, and you get rid of that bacteria, now what you're left with is yeast. And now that yeast is free from competition. They're no longer competing for resources. They're not competing for food and for space, etc. And so what you end up with is you end up with this overgrowth of the yeast, and that's going to be our candidiasis. And so taking a broad spectrum for females can come with a side effect of getting a yeast infection as a result of taking that broad spectrum. In the mouth, it can cause something that's referred to as thrush. That's a yeast infection in the mouth. So what you're seeing is, here's a tongue, and this tongue has these little patches. And these patches can be white or kind of yellow in appearance. Typically, the group that's most at risk for thrush would be babies. And so for a baby, let's say that this baby is either breastfeeding or bottle feeding. And so the baby is drinking milk. And if that milk kind of sits in the mouth and it has yeast there, then that yeast can start to grow and it causes this condition called thrush. Now, if you've ever seen this in babies, sometimes it can be difficult to kind of tell the difference, because if you've seen a baby that nurses, sometimes their tongue can have like a white patch appearance on it just from the milk that kind of sits there. But the key difference is that if you go to wipe it away, the white patches, if you go to wipe it away and it wipes away, it's milk. If it doesn't, that is thrush. And so if you end up with a baby that has thrush, that has this yeast infection in the mouth, the treatment typically is something called nystatin. This is an antifungal drug. And if the baby has thrush, you're going to put this nystatin in there multiple times a day. And that will help clear up that yeast infection in the mouth. Now, other groups that are susceptible to Candida albicans would be patients who are diabetic, right? So diabetics, because oftentimes if they are diabetic, they are immunocompromised, right? As a result of not regulating blood sugars properly, they also have a weakened immune system. Those with immunodeficiencies, so AIDS patients can also end up with thrush in their mouth. And again, it comes down to the fact that if you are immunocompromised, right, if you're immunocompromised, your immune system is not functioning properly. So in healthy individuals, right, Candida albicans is not often going to cause an effect. It's more of an opportunistic pathogen. It causes more of an effect in patients who are immunocompromised and their immune system cannot fight off those normal organisms that might be in the body normally. And so AIDS is where the immune system becomes compromised. Catheterized patients. So again, if you think of in the vaginal area, if you have a urinary catheter, you have a tube coming in from outside the body. Whenever you have a tube coming in, there is always a risk for an infection. And so a yeast infection could be a side effect. If we look at infants again are susceptible to Candida. Their immune systems are not functioning optimally yet. In the case of if they're bottle feeding, if those bottles are not cleaned properly, that can make it more likely that the baby ends up with thrush. And then lastly, individuals taking antimicrobial medications. So again, females who take a broad spectrum antibiotic. So yeast infections in the vaginal area or in the mouth, typically not life-threatening. However, if the infection becomes systemic, meaning that the organism now gets into the blood, it can lead to something called endocarditis. And this is a swelling of the heart chamber and valves. And if this happens, if you get this endocarditis, patients might only live for 3 to 4 days. So this could be potentially life-threatening. So next, we're going to look at a non-pathogenic yeast, meaning one that is not disease-causing. And so the organism that we're going to talk about is Saccharomyces cerevisiae. Saccharomyces cerevisiae is baker's or brewer's yeast. And this name, Saccharomyces cerevisiae, literally translates to sugar fungus that makes beer. That's what the name means. And so that tells you that this organism is able to take sugar and do fermentation to produce alcohol. So these organisms can do alcoholic fermentation. They are facultative anaerobes, so they grow aerobically, they grow anaerobically, but if grown anaerobically, they will switch to alcoholic fermentation. And so this yeast can be used to make beer, wine, and bread. And this type of fermentation is going to produce carbon dioxide, CO2, gas, and ethanol or alcohol. So the CO2 gas and ethanol. Now, if you think about bread, right? If you think about bread, does bread, when it's baked, contain alcohol? The answer is no. Because when you take the yeast and you're going to make bread, when you make the dough, the purpose of the yeast is that if they do fermentation and they produce that CO2 gas, that's going to cause the dough to rise. So it's going to cause the dough to rise. And so the purpose of the yeast for bread is to make the dough rise. The reason it doesn't have alcohol in it is that when you bake it, right, if you bake it, the alcohol is going to evaporate off at the temperature to bake the bread. If you were to eat the raw dough, that would likely contain alcohol in it. But once you bake it, it's no longer in there. And so if we look at Saccharomyces cerevisiae, you can see that on the plate, right, it looks like bacteria. Notice the colonies are smooth and round, and they're not fuzzy like a mold would be. And then if we were to look under the microscope and do a wet mount of this yeast, here you can see one that is undergoing budding. You can see the bigger mother cell and then the daughter cell beginning to bud off. There's actually a few of them that you can see this. And so this is a non-pathogenic yeast. And so this is showing you So this slide is just showing you what it would look like if we were to see this in class. And so in class, we would have a plate out, and that plate would have Saccharomyces cerevisiae, again, to demonstrate to you that the colonies that grow on the plate have a bacteria-like appearance. The colonies are smooth, they're shiny, etc. This is showing you on the right a simple stain, what we call a wet mount, of Saccharomyces cerevisiae. And so the way that we do this is that we take a drop of methylene blue and we put the methylene blue on a slide, and we actually use a toothpick and we pick up some of the yeast from this plate and we swirl that yeast in the methylene blue. And then we put a cover slip on and we view it under the microscope. And so this is with a total magnification of 400x. And so you can see that these cells are kind of round in appearance. And this is what you would see if you saw Saccharomyces cerevisiae. And so this is just to show you what it would look like. And so this is just another demonstration of a wet mount as well for Saccharomyces cerevisiae. Again, you can see these blue stained round cells. And so this would be an example of what Saccharomyces cerevisiae would look like under the microscope. So now we're going to move on and talk about our molds. And the first mold that we're going to talk about is Rhizopus stolonifer. And Rhizopus stolonifer is black bread mold. It's a type of mold that's found on bread. Now, we need to talk about some structures that you would find in a mold. So we have these hyphae. The hyphae are these filament structures. And the purpose of the hyphae is that they're there for nutrient absorption. They're there for the organism to absorb nutrients. They also have a structure called rhizoid. And rhizoids are there for nutrient absorption and also anchoring of the mold. You can think of rhizoids as being analogous to the roots of a plant, right? And so the roots of the plant have a lot of surface area to help with absorption of water. The rhizoids have a lot of surface area for absorption of nutrients, and it also helps to anchor the mold into its environment. Now, when we look at the hyphae can be divided into two main categories. And what that means is that if we have asepty, what that means is we have these cells with nuclei and there's no septum to divide them. So this is what we call asepty. A means without. It does not have a septum that divides the cells. And as a result, the hyphae are what we call coencetic. They are multinucleated cells. This is just one big cell with multiple nuclei. In a little bit, you're going to see that some molds have what is referred to as septae hyphae. And so here's the nucleus, here's the septum. Here's the nucleus, here's the septum. Nucleus, septum, so on and so forth. So there's this wall that is actually dividing the cell. But in organisms like Rhizopus, they have asepty hyphae. There's no septum that divides those two cells. And so we have an asepty hyphae. And then coming up from the hyphae is going to be a structure that's called the aerial hyphae. Aerial hyphae. So again, the way I like to think of this is I think of it like a plant. And the main hyphae and the rhizoids are like the roots. The aerial hyphae, aerial means it's going up to the air. Think of that like the stem of the plant, right? It's the part that's going to hold, if we're talking about a flowering plant, it's going to hold the flower up. So in the case of the molds, they have these aerial hyphae. And you're going to see in a minute that the purpose of the aerial hyphae is to hold the spores upright because the spores are used for reproduction. And so you're going to see that in a minute. The aerial hyphae in Rhizopus stolonifer is called a sporangiophore. PH. Okay, and so sporangiophore is the aerial hyphae. That is going to be in contrast in a minute. You're going to see what's called sporangiospores, which are the actual spores. That's different than the sporangiophore. The sporangiophore is the aerial hyphae. On the right here, you're seeing a tube that has some Rhizopus stolonifer in it. And so this is going to be your black bread mold. So if we look at Rhizopus and we look at the way that they can reproduce either asexually, meaning without a partner. And if they do that, they reproduce with a structure or spores that are called sporangiospores. So this structure that you're looking here is referred to as a sporangium. And the sporangium is an encapsulated structure where the sporangiospores are stored. So if we talk about sporangiospores, that type of spore is encapsulated. The structure that contains them is going to look smooth. You're going to see in a minute that that's in contrast to another type of spore that we're going to talk about. And another type of spore that we're going to talk about is what's called a conidiospore. And a conidiospore is going to be a naked spore. They're not encapsulated. Instead, they're just free spores. And you'll get to see the difference of that in a minute. So the sporangiospores are used for asexual reproduction. They are encapsulated within a sporangium. Here we have our sporangiophore. This is our aerial hyphae right here. And so again, that's going to hold those spore-containing structures upright. So this is our sporangiophore. One of the ways that you can tell that this is asexual versus sexual is if you look at the picture on the left, notice that to the structure that contains the spore, there's only one hyphae going to that structure. Only one hyphae. That tells you that there's no partner involved. This is asexual. That is in contrast to the picture on the right. The picture on the right is a type of reproduction that Rhizopus uses to reproduce sexually. And we call this a zygospore. A zygospore is used for sexual reproduction. It involves two partners. Notice we have two hyphae going to this zygospore. And so we have these opposite mating types, what's called a positive strain and a negative strain. And these two strains can come together to reproduce sexually, and they will form a structure called a zygospore. And so Rhizopus can reproduce either way. They can reproduce asexually using sporangiospores or sexually using a zygospore. So the next one that we're going to talk about, Penicillium notatum. And Penicillium notatum is a medically important mold. And it's medically important because Penicillium chrysogenum is a type of fungi that is used to produce penicillin antibiotic, right? And so penicillin antibiotic, you might recall, was discovered on accident by Alexander Fleming. He saw this fuzzy stuff growing on his plate, and he noticed that in the area where the fuzzy stuff was growing, that there was no bacterial growth. And so penicillin was discovered simply by accident. If we're looking at penicillium growing on a plate, what you're going to notice is that the edge of the colonies, the edge of these growths, are white, and the middle are green. The white are the hyphae, so those are the hyphae, and the green is going to be the spores. And so you'll notice that this outside part doesn't have spores yet because that's the part that's actively dividing, and it's making those groups of cells grow larger. And so you're going to see this white edge with this green middle. Now, in addition to penicillium being important to produce penicillin antibiotic, it has other uses as well, or other things associated with it. One is that penicillium can also cause a lung infection in immunocompromised patients. So one of the things that you're going to see is that immunocompromised patients are at risk for most fungal infections, meaning it puts them at a higher risk of contracting a fungal infection. So Penicillium notatum can cause a lung infection in immunocompromised patients. Some strains of penicillium are used to make blue cheese and brie cheese. So they're used to make cheese. Penicillium marniferi, this is dangerous, and this can cause a serious infection. We obviously don't use that one in the lab. The one that we would use would be the Penicillium notatum. So, couple things to look at if we're looking at penicillium under the microscope. So, the first is going to be the hyphae. The hyphae is going to be septate hyphae. And the septate hyphae is going to have this septum that divides the cells. So these hyphae are not coencetic. They're not multinucleated cells because they actually have this septum that is going to separate those cells. The aerial hyphae aerial hyphae in this case is referred to as a conidiophore. Again, the phore part of that is referring to the aerial hyphae. This is called a conidiophore because the spores that are used by Penicillium are called conidiospores. So again, you need to be careful and look at the end. Does it say spores? Spores are the reproductive structures. Or does it say phores? And the phore is going to be the aerial hyphae. Now, conidiospores are naked spores. Notice they're not encapsulated like Rhizopus was. For this one, it almost looks like little fingers, right? And these structures are these little fingers that come out, and the spores are naked and they're at the end of these fingers. The structure that holds the conidiospores is referred to as the phyalides. And so these phyalides are going to hold the conidiospores, and these conidiospores are used for asexual reproduction. Again, notice there's only one hyphae. There's not two hyphae coming together. So this is another type of asexual spores. So if the spores are conidiospores, then the aerial hyphae is called a conidiophore. If we were talking about Rhizopus, Rhizopus's spores are referred to as sporangiospores. So the aerial hyphae would be a sporangiophore. So the name of the spores goes with the name of the aerial hyphae. It's the end of the word that becomes different. Now, if we're looking at the mold growing on the plate, this mold is grown on a special type of agar. It's not just grown on simple nutrient agar. It's grown on what's called Sabarod's dextrose agar, SAB agar. There's a reason that we use this type of agar and don't use just simple nutrient agar. And the reason for this is that this has a low pH of between 5 and 6, which encourages fungal growth. And so you might recall that when we talked about bacteria, most bacteria are neutrophiles. They grow best at a neutral pH. In the case of fungi are more likely to grow at a more acidic pH. So they are acidophiles. So if we grow the mold at a slightly lower pH, that is going to allow the mold to grow on the agar, but not bacteria. And so this is used to help encourage fungal growth. So this is why we use this special type of agar. And so this, I believe, is a question in your question set. And so it's because of the pH. The pH is lower for Sabarod's dextrose agar. Oh, so there it is. So low pH of 5 or 6 encourages fungi to grow and not bacteria. So the next one that we're going to look at is Aspergillusniger. And this is a medically important black mold. And so if we look at Aspergillusniger, Niger, think of black, right? So the Aspergillusniger has yellow hyphae. So notice that the edge of those colonies is yellow. And they have black spores in the middle. And again, they have this very fuzzy appearance. So black spores, yellow hyphae. Aspergillusniger was named that because it was thought to resemble an aspergillum which is a holy water sprinkler in the Catholic Church. And so that's where the name comes from. This is one of the most common molds. It can be found in walls sometimes, and it causes a lung infection that's called aspergillosis. And so aspergillosis, again, is going to be more common in patients who are immunocompromised, as well as patients who have a variety of lung conditions, meaning it's more common in somebody who has tuberculosis or cystic fibrosis or asthma. This is an opportunistic pathogen. It is going to have a more of an effect on patients who have some pre-existing condition. In some cases, patients can cough up these fungal balls, but those fungal balls are not contagious. The disease is not passed from person to person. It's typically from the environment. So like if you're outside and you're, digging with a shovel and dust is coming up into the air, if there happens to be the mold spores in there and you happen to inhale it, well, then you could get aspergillosis. Or if you're working on a house, right, and you're doing demolition for a house, if you're not wearing a respirator to protect your lungs, and you're, let's say, knocking down walls, if there happens to be mold in those walls, you might be inhaling those spores. And if you inhale those spores from the wall and they go to your lungs, that's when it can cause this lung infection. Other species of aspergillus can be used to make soy sauce and soy paste. So there are other uses for this as well. If we look at what they look like under the microscope, so like Penicillium, they have septi-hyphae. So again, the hyphae has the septum and the nuclei are separated from one another. They have a conidiophore, that's the aerial hyphae, and they have conidiospores, which are these naked spores. So again, like Penicillium. This picture on the left is the picture that is in your lab book. The pictures on the right, the ones in the blue, are what ours look like when we do our staining technique. And so just be aware that ours looks a little bit different. It's a little bit more of a ball that you see. But notice how even though the structure is circular, right? Notice it looks similar to Rhizopus. The difference is if you look at the Rhizopus structure, the spores are encapsulated, meaning the edge is going to be smooth. In conidiospores, though, they're naked spores. So notice you can see these naked or these spores that are coming out. They're not encapsulated within a sporangium. These are conidiospores. And so these conidiospores, again, the aerial hyphae is referred to as a conidiophore. And so these are some examples of what they would look like under the microscope. So when we do this in lab, and I have a video to show you different methods to culture fungi, but when we do this in lab, normally our demo table, the table up in the front where you go to pick up materials, would have this covering on it, and we would soak it down with disinfectant. And we would tell you to only open the plates at the front table because we don't want those spores going all throughout the lab. Because if those spores are all over the lab and those spores happen to contaminate your bacterial plates, it might affect your result. And so typically we would keep our fungal plates up at the front table. And we would only work with them on the front table. We would make our slides on the front table, and then we would take the slides back to our desk and view them under the microscope. So what we would do for this is we would take a glass slide and we add 1 drop of what's called lactophenyl cotton blue. And so it's this blue stain, and the phenyl is going to kill the fungi. And so we add 1 drop of the lactophenol cotton blue onto the slide. And we don't want to touch the dropper to the slide because we don't want to contaminate the dropper. Then we have what we call fungal tape. And so it's basically like Scotch tape. It's just a clear tape. And for our lab, we don't use a whole piece. The whole piece is very big and it's not really necessary. So we would tell you to cut the fungal tape piece in half. You would peel off the tape and you would position it between your thumb and your middle finger. And you want to hold the tape in such a way that the sticky part is facing away from your hand, meaning that the sticky part is not facing your hand. It's facing away. You would put your index finger in between your pinky and your middle finger, and that index finger is on the other side of the tape. And that helps to then touch the tape to the plate where the fungal spores are. And again, this will make more sense when you see it in the video. So you will use gentle pressure with your index finger, and you press your tape onto the edge of the mold colony. So you don't want to aim for the middle. If you aim for the middle, you're not going to see those structures as much. You want to aim for the edge of that colony. So if I was looking at Penicillium, I would aim for where the green hits the white. That's going to give you the best structures. You don't want to take the older part of the culture, which is the middle. You want the newly dividing part, which is the edge. So you would take your tape to the outer edge, right where the spores and the hyphae meet, and you would push your tape down and you would put firm pressure. Now, if you don't push enough, you're not going to pick up those structures to see. If you push too hard, if you put too much pressure, you're going to smash down all of those structures, and you're going to lose what those structures look like. So it's a fine balance of pushing hard enough to pick up the structures, but not so hard that you're going to squish them. And so you are going to take your tape and you're going to put it on the edge of the colony and you're going to push down. Then you're going to take that tape and you're going to put it adhesive side down on the lactophenol cotton blue. And so you would take that tape and you would put it on the slide, and now you would have your spores underneath the tape with the lactophenol cotton blue. And so then you would look under the microscope and look to see if you see these structures. So this video is going to show you what this looks like for the lactophenol cotton blue. So this is going to show you how this works. So here is this video. So you can label your slide, and then notice what they're doing is they're going to pick up a little bit of the lactophenol cotton blue and put a drop on the slide. And so there's your lactophenol blue that's on your slide. And then you're going to acquire a piece of tape. And so notice that he's using, in his case, he's not putting his index finger in the middle, but the idea is the same. So you got to push down on the edge of that colony. And then you're going to take the tape and you're going to lay it over the lactophenol cotton blue. And when you do that, you're going to have this tape on the slide. And notice that for their tape is a little bit long. So they're going to cut off the edge with the scissors. And so you just want that tape to be on there. And then you're going to view that slide under the microscope. You want to make sure that there's no blue dye coming out the edge. So if you need to touch the edge with a chem wipe, that's what you would do. So when we do our tape mounts, one of the problems that can happen with doing a tape mount is that when we put pressure on the plate with the tape, that pressure pushing down to pick up those structures can sometimes, again, kind of squish those structures and make them appear different than what they actually look like when they're growing on the plate. And so a alternative to be able to view these structures is to do something called a slide culture. And what that involves, and I'm going to show you a video of how this works in just a minute, but you have this agar cube. And this agar cube is where you're going to inoculate your fungi, and you can actually see these structures growing within the agar under the microscope. And so let me show you how this works, because we have pictures that we're going to share with you that are both from a slide culture as well as from a tape mount. So let's look at how this technique works. And so this is just showing you what you need. And so what they're going to do is they're going to cut out a piece of Sabarod's dextrose agar into approximately a 1-inch square. And so they're using this knife to cut out a piece of this agar. Now, in this other glass dish to the left, you can see this U-shaped glass rod, and that is holding the slide. So you can see in there is the slide. That's where that piece of agar is going to go. So the person has picked up their Sabarod's dextrose agar, and they're going to transfer it onto their slide. So there they go. There's their agar. Now, the next part is going to involve inoculating the agar. And so they're using something called a spud. It's like a little right angle shaped, almost like needle. And so they picked up some of the culture with the spud. And notice that they're going to use the needle and place it just below the surface. So this one here is holding it in place, and the L shape, notice they're going to poke into the agar. So see, it's going into the agar a little bit below the surface. And so they're going to pick up more of the culture, and they're going to inoculate all 4 sides of the block a little bit below the surface. So she's picking up more using aseptic technique. And now going to inoculate another side. So poking into the agar each time as you go around. All right, so they've inoculated the agar. And then they're going to take the cover slip and cover the agar. And you gently want to push down on the cover slip so that it sticks to the agar. And the blotting paper in the bottom is going to be moistened using several drops of sterile distilled water. Because again, remember that molds like moisture. And so we want to get this environment to be moist, to be damp. And then this setup, once the lid is put back on, is going to be incubated at room temperature for the appropriate amount of time. And so that is going to be important because most molds like to grow at room temperature. And so these next set of slides are pictures from the lab that you would normally see in class. And so when you're studying these for your fungi, for your practical, you want to make sure to know what the mold looks like on the plate, right? So what does it look like on the plate? What does it look like with the lactophenyl cotton blue? So with the tape mount, and what would it look like on the slide culture? And so I'm going to walk through and show you pictures from our lab so you can see what this would actually look like. So the first one is going to be the rhizopus. And remember that the rhizopus is the black mold, right? The bread mold. And so it has this kind of fuzzy, it almost looks like pepper, like black and white appearance. And so that's what rhizopus looks like when grown on Sabarod's dextrose agar. Oops. If we look at it in a slide culture, right? And so notice here's the agar block right here, right? This bottom part. And then these structures are growing out from there. You can see that we have our sporangium. Notice that these balls of spores are smooth because they're encapsulated. Now, we're looking at this at 100x total magnification. You can't really go higher than this because the agar block, that agar cube, has some depth to it. And remember that when we use our 40x objective, that lens gets very close to the slide. Obviously, with this agar block on the slide, that doesn't really allow that to happen. And so these are not as magnified, but again, you can see this kind of smooth appearance, and that smooth appearance is the sporangium. And within the sporangium are the sporangiospores, right? The encapsulated spores. We have our, put this as a pointer. We have our aerial hyphae. This aerial hyphae, remember, is going to be called a sporangiophore. And so this is what the rhizopus would look like on a slide culture. Let's take a look and see what does this look like with a tape mount. So on the right is often what our structures would look like. Okay, and so here you can see, here is again the sporangiophore. This structure here is the sporangium, and within it are the sporangiospores. And so it's encapsulated. So that is the big way that you can distinguish between that and Aspergillus. Aspergillus is also going to have this round structure at the end, but you're going to be able to see individual spores along the edge. Rhizopus has a smooth edge because it's encapsulated. And so this is just showing you another example of what this would look like. But be aware that ours looks like the one on the right here. If you were to look at the rhizopus zygospores, remember that the zygospores are used for sexual reproduction. And so this is a slide from our sample. This is at 100x total magnification. Here is this zygospore. Notice that there's two hyphae coming to this spore structure, which tells you that this is sexual. One of those hyphae is the positive mating strain, and the other is going to be the negative mating strain. And so they come together to mate and to form this zygospore. If we look at Aspergillus, right? So Aspergillusniger, for example, remember that the edge of the colonies are going to be yellow. Those are the hyphae. And the black in the middle is going to be the spores. And you can see that fuzzy appearance that is very characteristic of molds. And so this is what Aspergillus looks like when growing on a plate. If we were to look at Aspergillus as a slide culture, you can see that we have those ball-like structures, again, similarly to rhizopus, but notice the edge of these structures is not smooth. They're dotted. It has those conidiospores, which are our naked spores. So again, This here, these would be my conidiospores. This aerial hyphae would be my conidiophore, right? So conidiophore. And so this is what Aspergillus would look like on a slide culture. This is Aspergillus tape mount. So again, this is what it would look like in our experiment in lab. So here again is going to be your conidiospores. Again, it's a round ball, but those spores are naked. They're not encapsulated. And so that's why those spores are referred to as conidiospores. This structure here, this is going to be our aerial hyphae, which is our conidiophore. And so we can see another example of that here. So this is the aerial hyphae. This is the main hyphae that's running perpendicular to it. And so this is what an Aspergillus tape mount would look like. And then lastly, we have our Penicillium. And remember that Penicillium is going to have white on the edge because the hyphae are white, but it has the green spores. So green spores, white hyphae. And so that's what Penicillium would look like grown on Sauberode's dextrose agar. If we look at the slide culture, right? If we look at the slide culture, here we can see, right? These are our conidiospores, the naked spores. They're being held up by the phyllides. And then this is the aerial hyphae, again, which is called your conidiophore, right? That's your aerial hyphae. And so this is just showing you what the Penicillium would look like using the slide culture. Here is what the Penicillium tape mount would look like. So this is a good one here. You can see the structures. So these are the conidiospores. These little parts here, this blue kind of line looking part, that's the phylline, which is holding the conidiospores. And then this is going to be your aerial hyphae, which is going to be your conidiophore. Now notice that for some of these, again, when you use a tape mount and you put pressure down, you're going to disrupt some of those structures. And so you can see some of these free spores that might simply just be to the way that we collect the specimen. Because when we put pressure down, we're likely to disrupt some of those structures. They're not going to look exactly how they would if they were actually on the plate. And so that's the advantage of having both a tape mount and a slide culture, because the slide culture is not going to be disrupted by preparing it that way. And so this is going to be your fungi.